Okay, my name's Russell Schwartz. Over the years, I've worked broadly on many areas of computational biology, but my lab's work can roughly be divided into two main areas: biophysical simulation and computational genetics and genomics. In the biophysics area, our main problem has been studying molecular self-assembly, which is a process by which various components, let's say proteins or other small molecules, come together to spontaneously assemble or disassemble various complexes, structures, and molecular machines to perform their functions within the cell. So biological research has been very heavily influenced by concepts of systems biology. That is, the idea that you can't understand biology just by looking at the components. You really need to know how they're acting collectively to perform some kind of function. And self-assembly is really crucial to that because it's one of the main ways biological systems act collectively in order to perform various functions within the cell. So I would argue that you really can't understand complex systems in biology if you don't have a good handle on self-assembly. And doing that is a lot of what my lab has been working on from the computational perspective. We've worked for many years on how you simulate complicated self-assembly processes. So simply the being able to get a handle on how you would efficiently and accurately model a complicated system like that, which presents many challenges you don't see in more classic biochemical systems. More recently, we've been focused specifically on the question of how to fit models to experimental data. So that is, how we would learn a model for a particular system, let's say the formation of a virus, and fit that model to experimental data that's available to us. And that's a very challenging problem for self-assembly because we don't have very good experimental methods for observing self-assembly in process. So we don't have any way of looking at a nanometer scale size assembly and watching each individual step of that assembly. So we have to rely on very indirect kinds of data and use simulations to figure out what simulation models, what properties of the system might be consistent with the experimental data we have. And then by learning the simulations that fit the data, use those simulations to infer missing pieces in the experimental system that we have no way of directly observing. In the genetic side, we've been very interested in questions arising from processes of evolution at various time scales. So that might mean evolution of individuals within a population like the human species, although for the past 10 years or so, our focus has been very specifically on evolution within cancers. So it's long been understood in cancer research that cancer is fundamentally an evolutionary system, that the growth and proliferation of cells within a tumor is described by the laws of evolution, so diversification and selection. But it's really only relatively recently that the field has come to understand that those principles are actually very important and necessary to understand the complexity of tumor genomic data we're now able to study in the laboratory, that we're now able to get our hands on. So my lab has been working on that question as well from the computational perspective, in part trying to characterize intratumor heterogeneity, which refers to variation cell to cell in single tumors, and in part to use this information about intratumor heterogeneity to build evolutionary models of how the individual cell populations in one tumor have evolved from an initial diseased cell to produce the tumor and eventually go on to progress and metastasize. In part, this is really a problem of basic cancer research. We're trying to understand how the rules of evolution apply to cancers and the many ways in which cancer evolution is very unlike other sorts of evolutionary processes people study. And in part, we're trying to understand how this process differs from patient to patient, because we know that the forces of diversification and selection are actually quite idiosyncratic by patient, and that this has important information for the treatment and outcomes of those patients. We're also trying to use this information to bring these evolutionary models closer to the clinic by looking at how we can study the evolutionary process of a tumor up to the time a doctor is seeing a patient in the clinic, and use that to infer what's likely to happen next to the tumor in that patient. This question of future progression of tumors is of enormous medical significance because currently there's a great deal of guesswork for the doctor in the clinic in trying to figure out what's likely to happen to a tumor next and what treatments, if any, are optimal for that specific patient. We've been able to show that features we can extract from inferred evolutionary models of single tumors give you a great deal of predictive power for figuring out how that tumor is likely to progress. For example, whether a primary tumor is likely to go on to metastasize.